20,000 years ago, during the relentless grip of the last glacial maximum, the North Atlantic was not merely an open ocean. It was a chaotic frontier of shifting sea ice extending from the coast of present-day France to the jagged edges of North America. It was a biologically sterile wasteland, a white void where temperatures plummeted far below freezing, and survival seemed mathematically impossible. Yet, a controversial theory suggests that amidst this frozen hellscape, bands of Stone Age Europeans paddled skin boats along the ice edge, hunting seals and defying the elements to become the first human inhabitants of the New World. Could the history of the Americas be fundamentally wrong? Did a lost tribe of Ice Age Europeans discover the continent 10,000 years before Christopher Columbus, only to vanish into the genetic mist? These questions sit at the center of one of the most volatile debates in modern archaeology: the Solutrean Hypothesis. For decades, this theory relied entirely on the silent testimony of stone. But it has recently collided with a far more precise and unforgiving witness: the molecular record of ancient DNA. The controversy begins not with bones, but with blades. In the solitudes of southwestern Europe, specifically France and Spain, a culture known as the Solutrean people flourished around 22,000 to 17,000 years ago. They were master craftsmen, renowned for creating the Laurel Leaf Point, a stone tool of exceptional beauty and lethality. Across the ocean, in the sediment layers of North America dated to roughly 13,000 years ago, archaeologists uncovered the Clovis Point, the signature weapon of the earliest undisputed Americans. To the untrained eye, they are just sharp rocks. To experts like Dennis Stanford of the Smithsonian, the similarities were undeniable and arguably impossible to ignore. The connection relies on a specific, sophisticated engineering principle known as Overshot flaking. This is not simple chipping. It involves high-velocity physics. The napper must strike a prepared platform on the edge of the stone with a billet, usually made of antler, at a precise angle and force. The goal is to send a controlled shock wave traveling completely across the face of the stone, removing a flake that reaches the opposite margin. When successful, this technique thins the biface dramatically without narrowing its width, creating a projectile that is incredibly flat, yet structurally sound for penetration. Historically, this technique is incredibly difficult to master and rare in the archaeological record. It appears in the Solutrean assemblages of Europe, and it appears again, thousands of years later, in the Clovis assemblages of North America. It does not, however, appear in the stone tool technologies of Northeast Asia or Siberia during the same period. Proponents of the Solutrean hypothesis argue that such complex engineering could not have been invented twice by accident.
That makes no sense.
They contend that the knowledge traveled across the Atlantic ice bridge, carried by Solutrean mariners who eventually morphed into the Clovis culture. However, the archaeological community remains deeply divided. Historians and lithic analysts argue whether this resemblance is the result of direct cultural transmission or simply convergent evolution. The idea that different groups of humans, facing similar needs for efficient hunting tools, independently unlock the same fracture mechanics. The skeptics point to a massive chronological gap. The Solutrean culture vanished in Europe nearly 5,000 years before the Clovis culture appeared in America. For decades, this stalemate persisted. It was a war of stones, with each side interpreting the flint and chert differently. But stone tools, for all their craftsmanship, are inanimate. They cannot speak to lineage, ancestry, or blood. To settle the debate, science required a biological time machine. That machine arrived with the sequencing of the human genome, shifting the battlefield from the shape of a knife to the sequence of a double helix. The question was no longer about how these people made their tools. but who their mothers were. To validate or debunk the possibility of an Ice Age Atlantic crossing, scientists turned away from the ambiguous shapes of stone tools and looked inward, to the cellular level. Specifically, they focused on a unique genetic structure located outside the nucleus of the cell, the mitochondria. This is the biological engine room of life, responsible for generating the chemical energy that powers every action, from a heartbeat to a thought. However, for geneticists, the mitochondrion possesses a distinct engineering feature that makes it an invaluable historical tracker. Unlike nuclear DNA, which is a shuffled deck of cards combining the genomes of both mother and father, mitochondrial DNA, mtDNA, is passed down exclusively from the mother to her offspring. It does not recombine, it does not dilute. It is a biological clone, copied and transmitted through the maternal line with near-perfect fidelity for millennia. Because of this lack of recombination, the only changes that occur are random mutations, tiny spelling errors in the genetic code that happen at a predictable rate over time. These mutations accumulate like ticks on a clock, allowing scientists to not only trace ancestry, but to estimate exactly when two populations separated. When researchers began sequencing the mitochondrial DNA of indigenous peoples across the Americas, they identified 5 primary maternal lineages, or haplogroups, that account for nearly all Native American ancestry. 4 of these, haplogroups A, B, C, and D, are undeniably Asian in origin. Their roots can be traced directly back to populations in Siberia and East Asia, supporting the mainstream theory of a migration across the Bering Land Bridge. But the 5th lineage was different. It was an anomaly that did not seem to fit the pattern. This was haplogroup X. Haplogroup X is a genetic outlier in the story of the Americas. While it appears in certain Native American populations, most notably the Ojibwe and other groups around the Great Lakes region,
it's not an error.
it is almost
it's the
entirely
source.
absent
It
in modern
changes
East Asia
everything we
and
thought we knew.
Siberia. Instead, the highest concentrations of haplogroup X in the Old World are found thousands of miles away. In the Middle East, the Caucasus, and among the Druze people of Israel, as well as in low frequencies across Europe and the Orkney Islands. For proponents of the Solutrean Hypothesis, this was the smoking gun. They pointed to the map and argued that the distribution was geographically impossible under the standard model. If the first Americans came solely from Siberia, why is there no haplogroup X in Siberia today? Why does a genetic marker found in the Great Lakes share a common ancestor with lineages in France and the Middle East? The Solutrean advocates proposed a direct solution. The carriers of haplogroup X did not walk across Asia. They paddled across the Atlantic. They argued that Solutrean women carrying this distinct European lineage arrived on the east coast of North America, bringing their specific stone tool technology with them. This would explain why haplogroup X is concentrated in the northeast of the continent rather than the northwest, and why it seems to bypass the Asian landmass entirely. However, the presence of a European-looking lineage in North America is not proof of migration. It is merely data requiring interpretation. Historians and population geneticists still argue fiercely over the interpretation of this absence. Does the lack of haplogroup X in modern Siberia prove it was never there? Or is it a case of genetic drift, a statistical phenomenon where rare lineages simply die out in a population over thousands of years, leaving no trace in the source region while flourishing in the new colony? To solve this riddle, scientists had to look deeper than the letter X. they had to examine the subtitles of the genetic code. They needed to pair the specific mutation markers of the Native American variety, known as X2A, against the European variety to see if they were truly mother and daughter, or merely distant cousins separated by vast stretches of time. The answer lies in the molecular clock. The superficial resemblance between a Solutrean blade and a Clovis point sparked the initial question, and the geographic anomaly of haplogroup X fueled the fire. However, in the realm of genetic genealogy, geography is secondary to chronology. To determine if a band of Ice Age Europeans truly crossed the Atlantic, scientists cannot simply look at where the DNA is found today. They must calculate when the lineages diverged. This requires the application of one of evolutionary biology's most powerful tools, the molecular clock. The engineering principle behind the molecular clock is based on the accumulation of neutral mutations. These are subtle changes in the genetic code that neither help nor harm the organism's survival. Because they are not subject to the pressures of natural selection, like a gene for thicker fur or sharper vision might be, they tend to occur at a relatively constant stochastic rate. Imagine a dripping faucet filling a bucket. If you know the rate of the drip and the volume of water in the bucket, you can calculate exactly how long the faucet has been running. In genetics, by counting the number of mutation differences between two populations, researchers can mathematically reconstruct the time to most recent common ancestor, to Marcel. When geneticists applied this clock to the specific Native American lineage, haplogroup X2A, and compared it to the European and Middle Eastern lineages, such as X2B or X2J, the results were devastating to the Solutrean hypothesis. If the Solutreans brought haplogroup X to America, the molecular clock should show a split occurring roughly 20,000 years ago, the height of the Solutrean era. Furthermore, the Native American X2A should look like a genetic daughter, or a very close derivative of the European X lineages present at that time. Instead, the sequencing revealed that X2A and the Old World forms are not parent and child. They are distant sisters. The molecular data indicates that the two lineages split from a common ancestor roughly 30,000 years ago, long before the Solutrean culture even existed in southwestern Europe. This temporal discrepancy breaks the chain of custody. The genetic evidence suggests that the ancestor of haplogroup X2A originated in the Near East, not Western Europe. From there, the population bifurcated. One branch moved west into Europe, eventually diversifying into the lineages seen in modern Europeans. The other branch, the one destined for the Americas, moved east. The crucial finding here is the sheer number of unique mutations found in Native American X2A that do not exist in any Old World population. For X2A to be so distinct, its ancestors must have been isolated for thousands of years, cut off from the European gene pool. This isolation allowed them to develop their own unique genetic barcode before they ever set foot in the Americas. This phenomenon supports the Beringian standstill hypothesis, rather than a rapid Atlantic crossing. The engineering of the genome tells us that X2A was not a passenger on a Solutrean boat. It was a silent traveler, trekking across the vast steppes of Eurasia. It survived the deep freeze not in France, but likely in a refuge in Beringia, the land bridge connecting Siberia and Alaska. While the stone tools of the Solutreans and Clovis people share a striking physical resemblance, the biological clock confirms that the hands that held them were separated by 10,000 years of divergent history. The stone is ambiguous, but the math of mutation is absolute. Yet, if the Solutreans are ruled out, a massive puzzle piece remains missing. If Native Americans are solely of East Asian descent, why do some of their genomes, specifically the Clovis people, show distant affinities to West Eurasians that contemporary East Asians do not? The answer would not be found in a living population, but in the frozen remains of a toddler buried in the Montana hillside. In 1968, near the Shields River in Montana, construction workers accidentally exposed a rock shelter that would become the most significant archaeological site for the Solutrean debate. Buried beneath a layer of limestone debris were over 100 stone tools, all stained with red ochre, a distinct ceremonial pigment. Amidst these artifacts lay the fragmentary skeletal remains of a male toddler, approximately 18 months old. This site, known as the Anzac I burial, remains the only confirmed Clovis grave ever discovered. For decades, the boy slept in silence. But in 2014, technology finally advanced enough to allow him to speak. The sequencing of the Anzac I genome required a feat of bioengineering. After more than 12,600 years in the ground, the DNA within the bone had degraded into microscopic fragments, contaminated by millennia of bacteria and environmental noise. Geneticists had to use advanced shotgun sequencing, a method that breaks the genome into millions of random pieces and then uses powerful algorithms to reassemble them by matching the fragments against a reference human genome. This process effectively reconstructs a shattered mosaic, turning dust into a readable biological blueprint. For the proponents of the Solutrean hypothesis, the Anzac boy represented the ultimate test case. Since he was buried with Clovis tools, he was undeniably a member of the Clovis culture. If the Solutrean theory was correct, if the makers of these tools were descendants of Ice Age Europeans, then the Anzac child's genome should have carried a distinct European signature. He should have plotted genetically close to the ancient hunter-gatherers of France, Spain, or Belgium. The stakes were absolute. The stone tools said, Europe, but the blood would reveal the truth. The results were devastating to the Atlantic crossing theory. When the team led by paleogeneticist Eske Willerslev mapped the Anzac genome, they found zero evidence of descent from the Solutreans or any other Western European population of that era. Instead, the boy's DNA revealed that he was fully part of the Asian lineage that migrated across Beringia. In fact, Anzac I is so central to the history of the continent that he shares a direct genetic affinity with nearly 80 of all living Native Americans in North and South America. He is, quite literally, the genetic grandfather of the hemisphere. Critically, the analysis explained why the confusion existed in the first place. The Anzac genome did contain a very ancient signal that links to West Eurasia, but it was not from the Solutreans. It was a basal connection, a deep, shared ancestry that predated the split between Europeans and Asians by tens of thousands of years. The engineering of population genetics allows scientists to distinguish between gene flow, recent mixing, which would support the Solutrean hypothesis, and shared drift, common ancestry from deep time. The Anzac boy showed shared drift, not recent contact. Historians and geneticists still debate the exact social structure of the Clovis people. Whether Anzac I represents a general population or a specific ruling lineage, but the question of his origin is settled. The boy buried with the red-stained tools was not a lost European mariner. He was a first American whose ancestors had walked from Siberia. Yet, the faint Western ghost signal in his DNA remained a puzzle. If his ancestors didn't meet Europeans in the Atlantic, where did they pick up a genetic signature that links them to the West? The answer lay buried halfway around the world, near the shores of Lake Baikal. The discovery that the Anzic child carried a faint genetic signal linking him to Western Eurasia was the final lifeline for Solutrean proponents. They argued that even if the boy was primarily Asian, this Western ghost signal proved that European mariners had integrated into the Native American gene pool. However, this lifeline was severed not in the Atlantic, but deep in the frozen heart of Siberia, with the sequencing of a 4-year-old boy known as MA1, discovered near the village of Malta, close to Lake Baikal in south-central Siberia. The boy lived approximately 24,000 years ago, during the height of the last glacial maximum. The site was a harsh, wind-swept tundra, characterized by subterranean dwellings constructed from mammoth bones and reindeer antlers. For decades, archaeologists studied the physical artifacts found there, noting artistic similarities to the Venus figurines of Europe. But in 2013, the genomic sequencing of the Malta boy revealed a population that had been entirely lost to history: the ancient North Eurasians, NE. The genomic engineering behind this discovery involves admixture analysis. By modeling the statistical drift of alleles, geneticists reconstructed the boy's heritage. The results were startling. MA1 was genetically related to modern-day Western Eurasians, Europeans, but had no affinity with modern East Asians. Yet crucially, he shared a massive amount of genetic drift with the Anzic boy and modern Native Americans. This revealed the missing link in the peopling of the Americas. The first Americans were not a simple offshoot of East Asians. Instead, they were the product of a prehistoric fusion. Roughly 20,000 to 25,000 years ago, a group of distinct East Asians moved north and encountered this ancient North Eurasian population, the people of Malta. These two distinct groups mixed, creating a new hybrid population, the ancestral Native Americans. This explains the why behind the confusing genetic data. Native Americans carry Western genetic markers, not because Solutreans crossed the Atlantic in boats, but because their ancestors absorbed a population of ancient Siberians who were distantly related to Europeans. The Western signal was carried across the Bering Land Bridge, packaged inside the genome of the ancient North Eurasians. The engineering of human migration is rarely a straight line. In this case, it was a convergence of two streams in the high Arctic. Historians and geneticists still debate the precise location of this admixture event, whether it happened near Lake Baikal or on the Bering Land Bridge itself, and the exact percentage of ANE ancestry in the first migrants, which is estimated to be between 14% and 38%. Nevertheless, the existence of the Malta boy solves the riddle of the European connection without requiring a single boat to cross the Atlantic. The ghost population of Siberia effectively erases the need for a Solutrean explanation. The shared ancestry is real, but the geography was wrong. The connection was overland through Asia, not overseas through the ice. With the biological record now complete, the Solutrean hypothesis is left with only its original pillar, the stone tools themselves. If the people didn't cross the ocean, how did two cultures separated by thousands of miles and thousands of years invent the exact same technology? The answer brings us back to the physics of the stone itself. If the genetic evidence is absolute, proving that the first Americans arrived via the Bering Land Bridge and shared no direct lineage with the Solutreans in Europe, one haunting question remains. How is it possible that two cultures, separated by an ocean and 5,000 years of silence, produced stone tools that are virtually identical? The answer lies not in a shared history of migration, but in the shared reality of physics. This phenomenon is known as convergent evolution. In biology, it explains why sharks, fish, and dolphins, mammals, both evolved streamlined, torpedo-shaped bodies. They are not related, but they face the same engineering problem, moving efficiently through a dense fluid. The laws of hydrodynamics force a specific shape upon them. The same principle applies to technology. When distinct human groups face identical survival challenges and work with the same raw materials, they often independently arrive at the exact same engineering solutions. The why behind the similarity of Solutrean and Clovis points is rooted in the mechanics of conchoidal fracture. Flint, chert, and obsidian are isotropic materials, meaning they have no internal grain and break uniformly in all directions. When a napper strikes the stone, the force travels through the material in a Hertzian cone, a shockwave that spreads out at a predictable angle. There are only a finite number of ways to thin a biface without breaking it. To create a projectile point that is thin enough to penetrate a mammoth's hide but durable enough to withstand impact, the napper must maximize the width-to-thickness ratio. Overshot flaking, the specific technique used by both Solutrean and Clovis nappers, is the most efficient solution to this specific physics problem. It acts as a structural optimization strategy. By sending a flake across the entire face of the stone, the craftsman removes maximum mass while preserving the structural center line. It is not a secret recipe passed down from master to apprentice across the Atlantic. It is a technological attractor. Just as cultures on both sides of the Atlantic independently invented the bow and arrow and the pyramid, the ancestors of the Clovis people reinvented the Overshot flake because it was the best engineering response to the lithic materials they found in the Americas. Historians and archaeologists continue to debate the cognitive leaps required for such innovation, arguing whether independent invention gives enough credit to human ingenuity, or if it relies too heavily on environmental determinism. However, the genetic verdict allows us to appreciate the Clovis artisans not as students of lost Europeans, but as master engineers in their own right, who unlocked the secrets of stone through centuries of trial and error in a new world. The Solutrean Hypothesis was a compelling narrative, a romantic vision of Ice Age explorers defying the Atlantic. However, the rigor of genetic genealogy has separated plausible fiction from verifiable history. Through the comparative case studies of haplogroup X2A, the ANZIC1 genome, and the Malta board, we have seen that the biological record is unbroken. The X lineage is an ancient sister, not a daughter of Europe. The Clovis child is fully Asian in origin, and the Western ghost signal is a legacy of the Siberian steppe, not the French coast. The true value of this scientific revelation lies in the restoration of indigenous history. By dismissing the Atlantic crossing theory, we are left with a reality that is far more impressive. The ancestors of Native Americans survived the isolation of Beringia, conquered the ice sheets, and populated two vast continents solely through their own resilience and adaptability. DNA does not just identify who we are. It honors the specific, unbroken struggles of those who came before. The stone tools were merely the technology of survival. The genes are the undeniable testament of the survivors themselves.